So, today I am going to start on hypersensitivity once again. In fact, this is the continuation of the earlier class. Just to recapitulate your memory, hypersensitive reactions are inflammatory reactions within the cell mediated and humoral systems that culminate in extensive tissue damage and even death. Just to reiterate the same thing, under normal circumstances, when we are invaded with pathogens, then our immune system, both cell mediated as well as humoral immune system, are able to mount an immune response by way of localized inflammatory reaction. Now, this helps in obliterating the pathogen. However, in certain instances, there can be a heightened immune response, which can culminate in extensive damage to the surrounding cells. This could also be systemic. The inflammation can be so much that death can ensue. Hypersensitive reactions, as I discussed last week, can be understood under 4 different categories: type 1 to type 4. Type 1, 2 and 3 are antibody mediated, whereas type 4 is because of T cells. Of the type 1 and 2, 3 which are antibody mediated, the first one, type 1, which I have covered in much detail, is mediated through IgE. Type 1, 2 and 3 reactions happen or occur shortly after the exposure to the antigen. It could be 2 to 30 minutes as in case of type 1, or 2 to 8 hours as could be with type 2 and 3. Type 4 hypersensitivity, however, which again I will say is cell mediated, takes 2 to 4 days. Now, I have already discussed, like I said, about type 1, but I will just go over the summary of what I spoke last time. Type 1 hypersensitivity is mediated by IgE, which gets bound to high affinity receptors on mast cells and basophils. Subsequently, when the allergen presents itself again in the body, then allergen induces cross-linking of the Fc receptors specific for IgE, which happens through the IgE which gets docked onto Fc eta R1, which triggers signaling for the release of pharmacologically active molecules such as histamine, serotonin and subsequently bradykinins, leukotrienes, etc. So, now having talked about type 1 hypersensitivity, and if you might remember that the signaling process itself is quite elaborate, is dependent on ITAMs, that is immunoreceptor tyrosine based activation motifs, and therefore, phosphorylation of several molecules in the mast cells and basophils, all of which in fact go to releasing first preformed granules from the cytoplasm and subsequently by way of allowing calcium to get into the cell. There are other activation processes which now makes the cell synthesize molecules like bradykinins and leukotrienes, which then get exocytosed. So, the primary response is milder than the secondary response in case of type 1 hypersensitivity when mast cells start getting degranulated. Now, let us come to type 2 hypersensitivity. Now, I had already mentioned that type 2 hypersensitivity is also antibody mediated, but this is not through IgE. So, let us look at what happens in type 2 hypersensitivity. Now, this is antibody mediated cytotoxicity, and the examples one can give are transfusion reactions, hemolytic disease of the newborn, drug-induced hemolytic anemia. Now, in all 3 cases, of course, these are responses which happen only under conditions where, for example, in the first case there is a transfusion reaction. Now, one cannot talk about transfusion reactions until one talks about the antigens which are present on red blood cells. Only then can one understand how these transfusion reactions can lead to cell mediated cytotoxicity. I have 3 pictures here, one which represents, now I am sure all of you know that by the method of classification of Landsteiner, which is now commonly used for typing blood groups in individuals, all of us can either be of type O, type A or type B. I am sure it is common knowledge now that individuals who are type O do not have any antigens either A or B on the RBC surface. People who have A type blood have the A antigen and also have in their circulation anti-B. That means antibodies, naturally occurring antibodies to antigen B. Similarly, individuals who are B, who are typed as B, have the B antigen and have in their bloodstream antibodies to A. Now, O group and people would have antibodies both to A and B, and there is a fourth class which of course is not mentioned here, which would be A plus B. So, there are individuals who have both A as well as B antigens and therefore, would not have any antibodies either to A or B. This therefore allows people with O group blood to donate blood to either A, B or AB, and AB individuals who can receive blood. Now, of course, the presence of antigen, since I have drawn this, maybe I should just discuss. There is a stem cell factor, first a protein or a lipid on which there are additions of sugars, and the sugars, the 4 chains that you see here, all of them are present on all the blood groups. You can see the 4 hexagon that are present on all O, A, B, and of course, it would also be A, B. Therefore, now the sugars N-acetyl galactosamine green, galactose followed by N-acetyl glucosamine followed by galactose. Now, these 4 and the last one is bound to fucose. Now, therefore, these 5 sugars in sequence are present on all the blood group red blood cells. Now, the addition of a terminal N-acetyl galactosamine determines that person to be having A antigen, and the termination of the chain with galactose determines the person to be of B antigen. Now, how is it that there are naturally occurring antibodies? Now, this is a question which is quite simple, but this is some answer to which very often people are not able to come up with. Now, initially of course, there are no naturally occurring antibodies in newborns. Now, soon after birth, one starts to get colonized with bacteria which are common cells. They are beneficial to us. Many of these bacterial antigens or sugars are similar to A and B, and therefore, once they colonize, and if the sugars are of A type or B type, they would start to evoke an immune response. Sugars, as you might remember from my previous lectures, sugars are weak and immunogens, and therefore, they only evoke a response which is restricted to being IgM. Now, why would an A antigen person have anti-B? That is simple. You have also learnt about clonal deletion of self-reactive antigens, self-antigen, which would mean that an individual in the bone marrow would have B cells that have the capacity to make anti-A and anti-B. But those B cells in A individuals, those B cells that have receptors for A, anti-A, making antibodies to A, would get automatically deleted, because in the bone marrow these immature B cells which have only IgM receptors, not IgD as yet, would get exposed to self antigen and undergo apoptosis and therefore cleared. Same would happen in case of B individuals. Now, in case of O antigen individuals, there would be neither A or B, and therefore, they have antibodies to both A and B. And of course, it therefore then plainly and simply follows that individuals with AB group antigens would have no antibodies to either A or B. So, if one has to use serum from individuals, one would be using serum from AB individuals, because they would have neither A nor B antibodies, and if cells are to be donated, then one would take cells from O group blood group people. Now, what happens now let us come back to this transfusion reactions. What would happen? I told you that there are naturally occurring antibodies in A individuals to B antibody, to B antigens. So, A individual, suppose an individual with A group gets by mistake now is given blood of B type, then the naturally occurring IgM antibodies to B would start agglutinating, and this would of course bring about antigen antibody interaction, small agglutinates. Red blood cells would get lysed because IgM antibodies are excellent for fixing complement. There would be complement, the classical pathway of complement mediated lysis would get initiated, and red blood cells would get lysed. There would be hemoglobin in the circulation, which of course can get thrown out of the system, but if there is too much, some of it can break down, before the hemoglobin is cleared, would break down to bilirubin. Bilirubin can be toxic if it crosses the blood brain barrier. Now, bilirubin is lipid soluble and therefore can cross the blood brain barrier, and deposition of it in the brain can cause problems. Now, in adults of course this does not happen, and transfusion reactions can be taken care of, are usually not fatal and can be taken care of as soon as the transfusion reactions can be seen by way of the person would suffer chills and fever because of too many red blood cells getting lysed in circulation, and also the split products being formed, which will also give rise to something similar to type 1 hypersensitivity. But as soon as the blood infusion or transfusion is stopped, these reactions will also stop. However, there are other conditions, hemolytic conditions, which could be fatal as happens in the newborn. Now, hemolytic disease of the newborn is the second that I would be discussing under this type 2 hypersensitivity. Hemolytic disease of the newborn, the one that immediately comes to one's mind would be because of Rh factor. Now, Rh factor, Rh is an antigen which is present on RBCs. Asian population, especially Indians, 70% of us are Rh positive, whereas in the West there is a larger population that is Rh negative. Now, Rh being a protein antigen, it evokes evokes an immune response by way of switching IgM type of response to IgG. If you might remember, while IgM cannot cross the placenta, cannot therefore go into the fetus, IgG has specific receptors on the placenta or trophoblast cells, because of which IgG can cross. Because of class switching, you might remember, there is also affinity maturation. So, whereas IgM antibodies might have lower affinity for Rh, once the IgM antibodies switch to IgG, there would also be automatic higher affinity antibodies. So, these IgG can cross the placenta and cause damage. Now, let us see what would happen in case of this erythroblastosis fetalis, because this is presented when Rh negative mother has an Rh positive baby. Now, the first child usually is safe because there is this blood barrier between the mother and the child, and only IgG antibodies can go through, and anyway the mother has not even made any antibodies to the Rh positive, because of this barrier the mother's immune system has not been exposed to the Rh factor. However, during birth, when the cord disengages itself from the uterine walls, there is some amount of mixing of blood that takes place. Some of the blood or the fetal red blood cells which are Rh positive now enter the circulation of the mother and now induces an immune response, which would be IgM to begin with, and would switch to IgG. So, the problem arises during the second child, when the mother is pregnant with the second baby. Now, if class switching has taken place and if IgG type of antibodies are made, then these would cross the placenta, enter the fetus, and now with a very efficient mechanism which is determined through through complement fixation, red blood cells get lysed. Now, this can happen in the fetus itself. This can lead, of course, to anemia, and therefore, ultimately if the reactivity is extremely severe, then it would lead to death of the fetus. Rh factor also, there are actually 4 different antigens, and I will not talk about them. But the factor D, this might ring a bell, that factor D in fact is the strongest of them all. And if one has an allele which is capital D, then antibodies that are made to this would definitely evoke a much stronger hypersensitive reaction than if it was small d, both of which would be recessive. So, there are questions, of course, people ask. There are cases where the mother is Rh negative, but yet, and all the babies are, where there could be as many as 3, and all the babies are Rh positive. How come there has been no problem? This only would be because of the allele of the stronger versus weaker type of Rh factors. Is there any treatment for this kind of case? Of course, yes. Now, the moment now of course, it is much simpler, one knows what blood group one belongs to, and a mother who is Rh negative, and if she is harboring an Rh positive, I mean if her husband is the woman's husband is Rh positive, and if they find that the fetus is Rh positive, then soon after the birth of the first child, the mother is given, I mean within 48 hours, the mother is given anti-Rh antibodies called ROGAM, and these antibodies lyse any fetal cells that are in the mother circulation, and you know that these antibodies would be very efficient in doing that, and thereby now the mother's B cells would not evoke an immune response at all, even before they become memory cells. Therefore, now the mother can, after this treatment of course, the mother can safely have the second child. Now, if one is talking about these Rh antibodies which are crossing the placenta, then there is a possibility that you can also have antibodies to the AB group individuals. Let us say for example, the mother is of the blood group B and she harbors a group baby. Now, under normal we have these naturally occurring antibodies which are IgM, which would be naturally occurring because of, I told you, triggering of the immune system of the person by way of bacterial antigens. Now, those would be only IgM type, but it has been seen that when fetal red blood cells go to the mother, then this kind of an immune response can, of course, will always be IgM to begin with, but can also switch to IgG. Fortunately, the immune response generated, even if it is IgG type to AB, it is mild, and this mild reaction as compared to Rh factor, which is a much stronger immunogen, antibodies to either A or B, even if they are IgG type, do not cause much harm. And you might have heard that there are babies born soon after birth, they have jaundice, and that jaundice can be taken care of very easily and it is not fatal at all. But nevertheless, like you to remember that even AB antigens can evoke a response similar but much weaker than Rh factor. Third type of type 2 hypersensitivity would be drug-induced hemolytic anemia. You might remember in another context of hypersensitivity, I did say that some antibiotics and small molecules bind to red blood cell surfaces. These serve as haptens. Penicillin, streptomycin, cephalosporin, these are very small molecules. On their own, they cannot evoke an immune response. However, when they bind to, some people's red blood cells are somehow more active with respect to conjugations. They conjugate penicillin, streptomycin, cephalosporin, these small molecules, and then thereby making these small molecules, haptens, immunogenic, these can evoke an immune response. There would be antibodies, and therefore, whenever a person has taken, is under treatment with these antibiotics and has made antibodies to these upon previous exposure, would now suffer from complement mediated lysis of RBCs. Now, as soon as this is realized, then of course, that particular antibiotic can be withdrawn, and this usually has never proved to be very fatal. So, we have seen type 1, we have seen type 2 hypersensitivity, both of which are driven by antibodies. Now, we look at type 3 hypersensitivity, which is also caused by antibodies. Let us look at that. Type 3 hypersensitivity, though caused by antibodies, this is after, if immune complexes are developed. Now, why should immune complexes cause any hypersensitivity? We do know that viruses, bacteria, any particulate antigens are very effectively thrown out of the system, are negated by the body because of the formation of immune complexes. A pathogen enters the body, there are antibodies made to these particles, antibodies, especially IgM and also of course IgG, can bring about cross linking of these antigens. Small complexes to large complexes are made, very effectively picked up by phagocytes and then deleted from the system. So, why should there be hypersensitivity? Now, this can only happen in unusual cases. For example, when there are antibodies present in the person, and now a large amount of antigen is injected. Let us look at what happens then. If you have too much of, there has to be a efficient and an optimum antigen antibody interaction which will give rise to large complexes. If you have too much of antigen, then much smaller complexes are formed, and if they are too many in number, then phagocytes are not adequate to clear the immune complexes from the system. So, what happens? They cannot be cleared, so they get deposited mostly along blood vessel walls, synovial membrane of joints, glomerular basement membrane of kidneys, choroid plexus of brain. So what if these complexes get deposited? What happens there then? What ensues is that these complexes are nothing but antigen antibody interaction, and if you remember complement mediated lysis, complement activation, then binding of the antibody to an antigen. If it is the antibodies of IgM or IgG type, then there is accessibility of the C1q binding site on the Fc region of antibodies. Now, of course, in circulation there are these complement components are in circulation. They get bound, the C1q gets bound to the Fc region or the region Fc, the second region, the constant domain of IgG and third of IgM, and then you have the entire cascade which we have already discussed. This brings about an inflammatory reaction in the way that split products are formed. You remember C3a, C5a and C4a. All these three have receptors on mast cells and basophils and neutrophils, and now these can activate the cells, mast cells, to degranulate and cause type 1 hypersensitive like reaction. Now, this is an inflammatory reaction. The other thing that can happen, of course, when the complement pathway is triggered, all the three pathways we know culminate in the formation of the membrane attack complex or MAC, and exactly that what happens. So, wherever there is site of deposition of these immune complexes, complement cascade has been initiated and the MAC that are formed, that are generated, can start attacking the sites where the complexes are formed. So, blood vessels get destroyed, synovial membrane, the membranes get destroyed, and glomerular basement membrane of kidneys also get destroyed. So, there is both inflammatory reaction as well as death of the tissue where the complexes get deposited. So, this could also be a chronic inflammation which persists for long periods of time. This type 3 reaction could either be localized type or generalized type. Let us look at the localized type. Now, for type 3 hypersensitive reaction to occur, there should be preformed antibodies in circulation, or yes, in circulation to that particular immunogen. Intradermal, if it is localized type, then it would be restricted to in what I am going to describe is on the skin. So, intradermal or subcutaneous injection of an antigen in animal that has high circulating antibodies to the antigen would now have a reaction at that particular site. And I will come to that in a little while. I have a picture which will tell you exactly what happens. But there are immune, because of the presence of high circulating antibodies and deposition of the antigen at one site, like I said, intradermal or subcutaneous, then there are immune complexes that are formed. This kind of a reaction is called Arthur's reaction, and the example of this condition is insect bite. Have you ever been bitten by an ant? Now, certain ants are able to evoke that small inflammation just around where the ant is bitten. Some of those that bite very, the bites hurt, actually do give much larger wheals. The other example is farmer's lung and pigeon fancier's disease. We'll come to that a little later. Now, there can also be generalized type 3 reaction. This is localized, generalized. So, here of course, the antigen, in this case the antigen is on a particular patch of skin. That is where, let us say, an insect has bitten, or in case of farmer's lung, it actually the antigen has gone and deposited in the alveoli. Now, generalized type reactions on the other hand is an example where there is a systemic disease. So, more than one particular site is involved, let us say serum sickness. Now, when would you think of serum sickness? What comes to my mind immediately is when a person has been bitten by, let us say, a poisonous snake, and the only antidote that is available right now in fact is horse serum, which is given to people who are bitten. The horse serum has antibodies to the entire snake venom. So, would have of course horse immunoglobulins. Now, there are antibodies. Now, this persists of course. Now, for the adequate protection from the venom, a large amount of antibody has to be given of the equine source, and this evokes an immune response. There would be antibodies to the horse immunoglobulins, and because there is large amount of antigen in this case, of course, is the horse immunoglobulins, and the other components of the horse serum, like even the albumin, which is seen as non-self, and a very strong immune response is generated, and this will of course form small complexes. Now, because the serum is given and it is in circulation, then of course one can imagine that this would be systemic. Autoimmune disease such as rheumatoid arthritis, systemic lupus erythematosus, Goodpasture syndrome, these also would evoke a generalized type 3 reaction, as well as in infectious diseases, meningitis, malaria, hepatitis. There are antibodies that are made to these antigens, to the pathogen, and whenever this pathogen number increases, you would have, or antigens increase, there would be generalized type 3 reaction. Now, you will be able to understand this type 3 reaction in the following way. Now, what I am going to describe to you is the localized one, where this is the Arthur's reaction, which can happen on the skin, and this one denotes what happens when an insect. Now, of course, this shows a syringe, but an insect has bitten and the venom has gotten in. Now, there are already antibodies to that venom, maybe because of a prior exposure, and has good amount of antibodies. Now, this is where the antigen enters. What you can see is little dots are the antigen, which are very strong antigens, of course. Now, because of the presence of immunoglobulins, there is a complex that is formed, but it continues to be present for some time in the skin before the complex is cleared. Before that, because of this antigen antibody interaction, complement cascade gets triggered and you have deposition of C3b at that particular site where antigen antibody interaction has taken place. C3b has receptors C3b binds to receptors CR1 which are present on neutrophils. So, neutrophils, when they come to that site and start now they get activated upon the CR1 receptor binding to C3b, and the neutrophils which have large amounts of lytic enzymes start pouring their enzymes. There are also another pathway here. When complement cascade is triggered, then you have the split products which are formed C3a, C4a and C5a. There are receptors for these split products of the complement cascade, complement components present on mast cells. This is a mast cell, and these are called anaphylatoxins because binding of these molecules to their cognate receptors induces signaling which is similar to what happens when allergen binds to the IgE specific antibodies which are docking onto Fc eta receptor 1. Degranulation therefore takes place of the mast cells. This now the histamine which is released and serotonin, they bind to histamine receptors which are present, if you might remember, H1 and H2 receptors which are present on blood cell the blood vessel wall, brings about now an activation which is similar to hypersensitive type 1 reaction. Now, therefore, you have now, because of the presence of neutrophils, large number of them which come to this site, this would definitely generate a inflammation at that site, and this is called an Arthus reaction. Remember, this is because of neutrophils. Now, it is only not neutrophils, but in Arthus reaction you will see mostly neutrophils. In case of the condition of farmer's lung, it has been seen that farmers who are constantly in contact with when they are on the farm and they are in contact with, let us say, birds, birds, the feces of birds very often when it's deposited, of course, the feces is deposited everywhere, but often this feces can dry and become airborne, and farmers get exposed to this. They inhale and these small particles now get deposited in their alveoli and evoke a response which is similar to this. So, these you can imagine that there is this inflammation in the lungs and accumulation of fluid. This could rather be debilitating if not fatal. Let us come to the type 4 delayed type hypersensitive reaction. Delayed type hypersensitive reaction, as mentioned earlier, the name itself suggests that after exposure to the antigen, the reaction happens 48 to 72 hours later, and this is mostly due to T cells. Let us see what happens in case of delayed type hypersensitive reaction. The example of this is reaction to the example is when individuals, this is a time tested analysis or diagnostic test which allows one to see whether one is infected with M. tuberculosis. Now, this test involves the antigen from bacteria PPD purified protein derivative. It is an extract of the bacteria M. tuberculosis, and this is put onto skin. This is a skin test. Now, if one has antibodies to the T cells to mycobacteria, and if the individual now is exposed to PPD, there is development of a red swollen firm lesion within 48 to 72 hours. So, this indicates a previous exposure in that individual to M. tuberculosis. Now, what is this lesion due to? The hard lesion, which is 48 to 72 hours, this lesion is due to intense infiltration of cells, 90% of which are macrophages at that particular site. One can see this delayed type hypersensitive reaction in people who in experimental animals. Most of us probably have at some point of time generated antibodies to different antigens of interest, and if you have done that, you would have used, commonly people use Franz complete adjuvant as the sensitization adjuvant. Now, Franz complete adjuvant has, apart from mineral oil as well as the detergent, which will allow an emulsification or a stable emulsion of the aqueous and the mineral oil, these two phases, also has heat killed mycobacteria. Now, this would of course have PPD, which would evoke this delayed type hypersensitive reaction, or activation of Th1 cells. So, when we normally inject the emulsion subcutaneously or intradermally, subcutaneously in mice and intradermally in rabbits, and within 72 hours, if you go and see the area where the antigen has been deposited, you will see a lesion. To begin with, it looks like there is some pus, but soon after it becomes a hard lesion. And if you were to take a slice of this, a thin section of this through this lesion, there will be, you will see that there is intense infiltration of cells, which are definitely different from the epithelia, and these would be macrophages. Now, here this particular slide tells you a little bit more in detail with the actual reaction that takes place. Now, the example I had given you, of course, is Mycobacterium tuberculosis and the immunization experiments that one can when one utilizes Franz adjuvant or complete adjuvant. But let us look at another condition of delayed type hypersensitive reaction, where there are small molecules which could be plant derived, for example, or this could be even a cosmetic. You might remember that there are people who are allergic to hair dyes or certain cosmetics like facial cream. That is because there are small molecules that are present, and not all individuals. Some individuals seem to react. How do they react to these? As the example over here is poison oak, but this could be a small molecule which could be, it could be turpentine. Some people are hypersensitive to turpentine. That is also a small molecule, just like this poison oak, which the toxin part of which is pentadecacatechol. This is a small molecule on its own, it does not evoke any immune response. But when pentadecacatechol binds to proteins of the skin, now this becomes a hapten, and this is taken in by the protein, as usually happens, would be taken by the antigen presenting cells which are present in the skin, Langerhans cell. These are efficient antigen presenters. They would take the entire protein, the skin protein, which has been modified now because of binding to pentadecacatechol, and the molecule now is presented in the context of class 2 molecules, APCs. So, class 2 molecules, and therefore, they present the antigen to Th cells, the helper cells. The Th1 cells get sensitized by this, and they start secreting gamma interferon as well as macrophage monocyte chemotactic factors. So, what the chemotactic factors do? They now send the message to, or they signal to blood monocytes to come to the site where the sensitization has taken place. And therefore, now there is a reaction, inflammatory reaction that takes place here. Delayed type hypersensitive reactions can be treated by just withdrawing the source of the molecule. Now, in case of poison oak, I mean, one would of course come across that only in a forest, for example. Now, in case of people who are sensitive to hair dyes, for example, or turpentine, then turpentine of course is an occupational hazard. So, you change your job, or if one is hypersensitive to hair dyes, then you stop using the hair dye, or to any of these cosmetics. Now, of course, there are cosmetics available which say hyposensitized, which would mean that the cosmetics do not contain allergic substances. Now, the last two, the type 3 and type 4, the localized type, the Arthur's reaction as well as the delayed type hypersensitive reaction look very similar. Both of them at the site, the localized one at the site of antigen deposition, similarly at the site of antigen deposition, but I would just like to tell you the two differences. One is because of about at the site of antigen deposition, large number, more than 80% are neutrophils in case of Arthur's reaction, whereas in case of the delayed type hypersensitive reaction, these are monocytes. Now, the type of cytokines that are secreted in these two instances would be different, whereas Arthur's reaction also engages mast cells for this inflammatory reaction. In case of delayed type hypersensitive reaction, it is totally by Th1 cells, delayed type hypersensitive cells. Now, you have Th1 response also of course in case of patients infected with and are diseased with lepromatous leprosy or with tuberculosis lesions. The reactions would be very, very similar. What happens in those cases is that there would be cells which are infected, like macrophages which are infected with bacteria. The Th1 cells get sensitized, but they cannot do anything to the bacteria which are sitting inside, and thereby they are all the time being activated. They keep on making cytokines which are deleterious if there are too much of it locally. So, I just like to summarize this part of my talk. We have come to the end of hypersensitive reactions. Now, I have already talked about type 1 earlier. So, let us just summarize type 2, 3 and 4. So, type 2 hypersensitivity happens when antibodies bind to cell surface antigens, activate complement, which brings about lysis of cells. Now, type 2 hypersensitivity all of these would be like type 2 hypersensitivity. The example is transfusion reaction or erythroblastosis fetalis. There is toxicity to cells and thereby lysis, and this is determined by antibodies. In case of type 3 hypersensitivity, there is formation of immune complexes. There are too many of them to get cleared. This therefore, the complexes get deposited in the body, they activate complement. The activation of complement, which is now it becomes almost a chronic kind of situation where there is tissue damage, split products affect vasodilation and accumulation of neutrophils at that site. Type 4 hypersensitivity is antigen induced sensitization of Th1 cells. This induces release of cytokines and accumulation of monocytes. You would agree with me therefore that none of these reactions would happen under normal circumstances. It would only be when in situations where the hypersensitivity is evoked because of conditions which normally would not happen. In case of IgE, normal conditions, normal individuals do not make too much IgE. Only a few people make too much IgE which gets sequestered onto mast cells and basophils. In type 2 hypersensitivity, it is only when by mistake one is given a blood group which is incompatible. Type 3 hypersensitivity is, well, you are bitten by a snake and you have to take the antidote to the venom, and if you have had to do that more than once, then you are surely going to get type 3 hypersensitivity. Type 4 hypersensitivity, well, examples what I have given you is turpentine or poison oak or cosmetics, and these are also whatever you say is artificial conditions. So, hypersensitive reactions are those pro-inflammatory, inflammatory reactions which are able to induce an unwanted kind of an immune response, okay, and this could lead to tissue damage. Now, let us switch gears and come to, let me introduce to you tolerance and autoimmunity. We have talked about hypersensitivity, which is too much of a bad kind of an immune response, which of course deletes the pathogen, but also causes tissue damage to the host. So, let us come now from hypersensitivity to autoimmunity. Autoimmunity, why should there be autoimmunity? We have always been talking about the immune system which is capable of recognizing self from non-self and therefore is able to mount an immune response only to delete the non-self. But it was early part of the 20th century that a scientist, Paul Ehrlich, realized that one may evoke an immune reaction against self antigens, and he called it horrid autotoxicosis, a condition which is deleterious to oneself, and the immune response is generated to oneself. Now, what are the diseases which come under this category of autoimmunity? Diabetes type 1, rheumatoid arthritis, systemic lupus erythematosus, Graves' disease, all these result from the failure of the immune system to distinguish self from non-self. Now, why would we make or evoke an immune response to self? By now, you have of course heard from me that self-reactive B cells are deleted in the bone marrow when those cells which have only IgM receptor on the cell surface are yet to also express IgD. When such cells, immature B cells, are exposed to antigens, whichever antigen, under normal circumstances it would be only self antigens, then the cells undergo apoptosis and therefore clonally deleted. So, this is in fact the central tolerance. Okay. So, there is central tolerance as well as peripheral tolerance, because of which we react normally to non-self antigens. Looking at self tolerance, this is a dominant mechanism which happens in the bone marrow in case of B cells and thymus, the medulla of the thymus in case of T cells. Now, though deletion of self reacting clones occurs, not all self antigens are expressed in the primary lymphoid organs where negative selection takes place. Now, of course, not all antigens are expressed in the primary lymphoid organs, but the entire immune system is sort of a network, and there would be migration of antigens through the circulation. Bone marrow is of course a part of the circulation. So, therefore, antigens, large number of antigens are in fact exposed to cells, or other cells, immature cells are exposed to a large number of self antigens, but not all self antigens are expressed in the primary lymphoid organ where negative selection takes place. So, those self antigens, small number of self antigen binding B cells would definitely escape and come into circulation. Also, it has been seen that there is a threshold of affinity for self antigens before clonal deletion can take place. Thus, some weakly reactive clones may persist in circulation. Fortunately, in spite of these small, this population of cells which are not destroyed come into circulation, fortunately we have a peripheral tolerance. which actually inactivates self-reactive clones. Now, how does that happen? This can be given by one example. Now people have made transgenic animals which have anti-HEL. What is HEL? Hen egg lysozyme. Now, hen egg lysozyme has been a popular molecule with immunologists. This is a molecule which has 4 disulfide bonds, and it's been studied very nicely with respect to B cell epitopes, with respect to continuous epitopes, discontinuous epitopes. For describing these type of epitopes, this molecule has been extremely useful. So, now there is a transgenic mouse that has been made to anti-HEL. So, a large number of B cells would have receptors for binding to specifically to HEL. Now, in these animals it has been seen that when HEL, now the HEL of course hen egg lysozyme would be a artificial system in case of mice. Now, the mouse has been made transgenic with respect to HEL. Normally we would have a very small number of each, a receptor bearing B cells to each antigens. Remember, we are supposed to evoke an immune response. We are capable of evoking an immune response to something like 100 million different antigens. Therefore, now if you want to study a particular population, you would make transgenic animals. This particular mouse now, which is transgenic, would have at least about, let us say, 33 to 40% of the B cells which react or have the receptor to react to HEL. Now, they studied afterwards, they have studied now how many of these HEL reactive clones get destroyed when these B cells in the immature state are exposed to membrane bound HEL versus soluble HEL. It was interesting that membrane bound HEL would all, that means another cell which is exposed, which is, again this would be an artificial system of course, has HEL. So, the HEL is fixed onto the membrane of another cell. Now, when HEL B cells are exposed to membrane bound HEL, almost all the clones got deleted. There were apoptosis seen. However, when such transgenic animals were exposed to soluble HEL, it was seen that quite a large population of self-reactive, now it would be self-reactive clones here, because this is a transgenic animal, self-reactive clones now come into circulation. But interestingly, in the circulation, when now transgenic animals were exposed to HEL, it did not induce clonal proliferation of the HEL bearing, anti-HEL bearing B cell receptor. So, therefore, we know that there is something known as peripheral tolerance, and unless this tolerance is broken, self-reactive clones continue to be in circulation, incapable of reacting to self, and we will understand in the next lecture how this barrier is broken. Another part of the peripheral tolerance, which I am not going to deal with, but it is important. This is going to be dealt with people who are Professor Nandi or Professor Manjunath, who deal with T regulatory cells. Now, I will just introduce to you this autoimmune diseases, and I will just, the details of which we will continue in the next lecture. But now, autoimmune diseases, we know now that there are autoimmune diseases. We can make antibodies or reactive T cells to different either organs or self molecules, or we will come to those. Autoimmune diseases can be studied under organ specific autoimmune diseases versus systemic. Organ specific immune diseases, where the immune response is targeted to a unique antigen which is specific for a target cell or organ, and therefore, the immune response causes direct cellular damage to that particular organ. The examples of these are Hashimoto thyroiditis, autoimmune anemia, Good-Pasture syndrome, and insulin dependent diabetes mellitus. Now, if you might, now of course, Hashimoto thyroiditis would definitely involve thyroid gland, autoimmune anemia would involve red blood cells, Good-Pasture syndrome involves kidney, and insulin dependent diabetes mellitus, of course, as the name suggests, would involve beta cells. Now, before I think I will stop here we will talk about organ specific autoimmune diseases in the next class, and we will deal with what are these antigens. Now, of course, we should remember with respect to organ specific immune diseases that of course now there would be small number of B cells which have escaped deletion, just because let us say in case of Hashimoto's thyroiditis. Now, those cells bearing receptors for the very antigens which later on are involved in disease progression. Now, these B cells have probably just were allowed to come into circulation, because they were not, the immune cells, immature immune B cells were not exposed adequately to enough amount of the thyroid antigens. Therefore, these have come into circulation. Again, under normal circumstances, there would be, these cells would not even be exposed to the thyroid antigens again, because the thyroid antigen is localized. This can only happen in situations where, let us say, there has been, something like molecular mimicry, which we are going to talk about, or there could be some kind of damage because of which to the thyroid gland, let us say, because of which a lot of cells have been destroyed, and therefore, these antigens have come into circulation. And just because there is large amount of the antigen that come into circulation, those quiescent self reactive clones in the peripheral circulation now get exposed and start to get activated. Insulin dependent diabetes mellitus, the same thing. Why should one make antibodies or T cells reactive to beta cells? So, all this we will be able to address in the next class, and we will start off with description of all the 4 diseases: Hashimoto's, autoimmune anemia, Goodpasture syndrome, IDDM, insulin dependent diabetes mellitus, and then go on to the systemic autoimmune diseases. Also, next class I will be talking about what are the mechanisms of these autoimmune diseases. But before I end, I would like to also tell you, there are reasons for which I am also going to explain in the next class, but there are reasons for which more women are prone to autoimmune diseases than men. So, obviously, steroid hormones must be playing a role. Thank you.